This is a tutorial for how to use Merizo-search on the PSIPRED web server. Merizo-search is a program designed for segmentation and then matching of protein structures on a per-domain basis. Before we get into how to use Merizo-search on the PSIPRED web server, it might be instructive for us to take a look at what the program actually does. This is a figure from the manuscript describing the method, and it shows the overall procedure followed by Merizo-search. The program consists of two steps. The first is that it takes a multi-domain protein structure that you provide as a query. It then segments it using a program called Merizo, which uses deep learning-based segmentation. And it converts this structure into regions called domains. It essentially takes that structure and labels each residue as belonging to one of any number of domains. And then once these domains have been identified, we pass them through another neural network model called Foldclass. And Foldclass converts single domain structures into what are called embeddings. Embeddings are simply vectors of numbers or arrays of numbers. And these are 128 long. And each domain is associated with an array of 128 numbers. The reason we do this is because it makes it quite easy to search very large databases of such domain structures. And that's shown here at the bottom, where we take a collection of single domain structures, we pass them through the same Foldclass neural network, and we compile a very large database of embeddings. And in contrast to matching with complete structures, matching embeddings in this fashion is incredibly fast. And this gives us the ability to search very large databases of structures in reasonable time. Once we have conducted that search, we can then rank these hits in terms of their so-called embedding similarity. We use what's called cosine similarity for these embeddings. And then we retrieve the top Ks. In the case of the PSIPRED web server, we retrieve the top 10 hits. And then because this is an approximate sort of matching, we're not conducting true structural alignment. Once we've identified those top K hits, we validate them using TM-align, which does carry out actual structural alignment before we call things as hits. And this allows us to do two things. One is to identify hits on a per domain basis. So given a multi-domain query structure, we can say, okay, this is where the domains are in that structure, and these are what those domains look like to domains that we've already seen in the PDB. But it also allows us to do certain other things. It allows us to conduct matching of entire chains using per domain matchings, but also being invariant to things like repositioning of the domains in 3D space, or indeed their order in the sequence. So this is another figure from our paper, and it shows that given a query chain that's comprised of three domains, green, blue, and yellow, we can obtain different categories of hits. So the most obvious one would be number 4, where the exact multi-domain architecture, that is the order and exact sequence and identity of the domains, is matched. We can also have contiguous domain matches where we allow any hit chain to have extra domains at either the N or the C terminus. We can then have discontiguous domain matches where the order of our query domains is preserved, but we can have optionally interrupting domains or additional domains in the hit chain. And then the last most general category of hit is called unordered domain matches, where the same domains are present but in any order and optionally with additional domains in the hit. So this is a very powerful and flexible way of searching multi-domain query structures against a database. So here in the bottom left, we show an example of a case where we are able to find a hit chain in which the exact same domains are found, except that one of the domains has been positioned in a completely different spatial location relative to the others. This is a type of hit that would be very difficult to find using a simpler comparison method that just uses the full chain structure. And so this is a unique kind of feature of the method. It also allows us to detect, as I mentioned, different orderings of the domains. So here in subfigure C, we have a query structure at the top which has 4 domains in it, in this order from N to C terminus. And we're able to match it against this other chain that has the same 4 domains in basically the same overall fold, but in a different order. In this case, it's a circular permutation. And as you can see, we've aligned these two structures on this sort of tin barrel domain in the center. And you can see that the accessory domains around it are placed in different positions. And again, this is the sort of match that would be quite difficult to detect using simple sequence-based matching or even methods such as fold-seek that operate on complete structures. Returning now to the PSIPRED web server, we'll show you how to run a Merizo-search job. Because Merizo-search takes in a PDB structure as its input, you have to click on the PDB structure data tab, select Merizo-search, and then choose your PDB file. I'm going to choose this one called 3BMV. This is a structure taken from the PDB. Please note that the structure has to be provided in PDB format. We do not currently support MMSIF, but that's hopefully something that we'll be adding fairly soon. So we click that, and with all predictors that take structure as input, it's important to specify which chain in the structure we want to search. In this case, we want to search chain A, so that can be left as is. On the web server, we give you the option of searching two databases. One is CATH 4.3, and this is a curated database of domains, all of which are derived from PDB structures. There are over 500,000 domain structures in CATH 4.3. So it's a good spread. It captures most of what's available in the PDB. We also give you the option to search TED. TED stands for the Encyclopedia of Domains. It is an attempt to automatically segment all 214 million AlphaFold database structures into domains. And this follows a different sort of segmentation and classification procedure as CATH does, but it is designed to produce domains that can be mapped to the CATH hierarchy. And we're able to do that for a vast majority of cases. This database is considerably larger than CATH, but can still be searched in a reasonable time frame using the PSIPRED web server. TED contains over 365 million domains segmented from AlphaFold database structures. So for the purpose, for the first run of this tutorial, we'll just use CATH. And because this is a PDB structure, it is already present in CATH, and so we have certain expectations of what the results might look like. So we'll give this job a name. We'll say test 3 BMVA CATH. And optionally, you can specify an email address to where you want the results sent. And once you've provided all of that information, you can click submit. And we'll wait for the results to appear. Okay, looks like our results are ready. I'll go through each of the panels on this results page, and then we'll take a look at the individual files that are available. So the first panel shows a visual representation of the domain segmentation that Merizo has conducted. Here it just colors different segments of your input protein structure according to where Merizo thinks the domains are. It's a good idea at this point to visually inspect this segmentation to make sure that you think it's a good segmentation. Segmenting multi-domain structures into domains can sometimes be challenging. It can be quite ambiguous as well. So this is a relatively simple case. Although it has 4 domains, it does look like the 4 domains have been identified correctly, and so we can proceed. The next panel lists what's called best hits, and this is a listing of the top 1 hit for each of the identified domains in the structure. So these are matches at the domain level, and we just identify the top 1 for a quick summary. This table allows you to quickly identify which domain we're talking about. So if you click the show button in any of the rows, it will highlight just that domain on the top structure. The chopping column tells you which residues in your query structure belong to this domain. So in this case, it's residues 15 to 398. The next column is called PLDDT, although it should probably read PLDDT/B factor, because this is essentially the average value in the B factor column of the PDB file over the residues in the chopping column. So if your query was an AlphaFold model, for example, this would in fact be the PLDDT of that domain. But because we've used a PDB structure as the query, this is just the average B factor. The next column is hit CATH, and this lists the CATH domain that best hits the particular selected domain. As we submitted a PDB structure 3BMV, you might expect that the top hits for each of the domains in this structure should also be from 3BMV. And indeed, we can see that for 3 out of the 4 domains, but not the first one. And this is because although the segmentation here looks pretty good, it doesn't exactly match what CATH has segmented this the same structure into. And because of that, there can be small differences in the embedding vectors and therefore small differences in terms of cosine similarity. And so, although we can see that this domain is probably a good hit, as we'll see shortly, it's not the exact hit, but it's still pretty close. The next column lists the CATH H family, that is the homologous superfamily that the hit belongs to. And this, both of these columns are links to pages on the CATH website. So we'll take a look at this one first. This is the website for the specific domain that we hit, and you can view the parameters of its classification. I won't go into too much detail on the contents of the CATH website pages, but it's a useful sort of link out into the CATH resource. The next one will be a link for the homologous superfamily that the hit is drawn from, and this website, this page will give you considerably more information because it's describing an entire superfamily of domains rather than one specific one. Again, I won't go into too much detail here, except to say that each homologous superfamily is further subdivided into what are called structural clusters and functional families, and these are really useful when you're trying to conduct evolutionary analysis of your protein. Okay, so the next column is hit PDB, and this is a link to the RCSB page for the hit structure that contains the domain that we've hit. So in this case, we can see that it's cyclodextrin gluconeotransferase. My query is here on this page, 3BMV. It is a cyclodextrin glycosyl transferase. So actually sounds fairly similar to my query, which is encouraging. Okay, returning to the results page, the next column lists cosine similarity, and this is the raw similarity of the embedding vectors that were used to call this a hit. This number goes from 0 to 1, with 1 indicating an exact match. And so as you can see for the subsequent domains, these values are very high indeed, meaning that we were able to match this very confidently to something in the PDB. And here the number is 0.88, and again, it's quite high, but there isn't a sort of direct mapping between cosine similarity and actual structural similarity. It's just that higher values in general are better than lower values. And this is one reason why we perform TM align matches and alignments to verify any of the hits that we find. Okay, so looking at the next columns, you have query length and hit length. And these are simply the number of residues found in the query and the hit domains. And again, ideally, these should be quite similar. But again, it's possible that you can have insertions and deletions in either of these domains relative to the other. So it's also useful to look at alignment length. This is reported by TM align, and it tells you how many residues could be aligned between these two structures. And as you can see, the alignment covers a good fraction of both of the query and the hit, and so that's a really strong alignment. Sequence identity is also reported by TM align, and this tells you in the range 0 to 1 what fraction of the residues match between the query and the hit domain. And here we have 74.3%, which is quite good considering protein domains. But for the others, it's 1.0, which means 100% of the residues match exactly. The next column is max TM, which is also reported by TM align. TM align will output 2 TM score values, one normalized by the length of the query and the other normalized by the length of the hit. We report the maximum of those 2 values, and this is again a number that goes from 0 to 1, with 1 indicating an exact match. So any number above 0.5 is considered to be a significant match in the sense that 2 domain structures will probably have the same fold. We apply a filter in our TM align runs of only reporting structures with 0.5 or more. And so anything listed here essentially is going to be a fold level match. So here we can see that all of our TM align values are quite high. Again, we have exact matches for 3 out of the 4 domains. So that's really good. The final column is RMSD, root mean squared deviation, which again is computed on the structures. Here, lower values are better. And if you have a value of 0, as we have for 3 of the 4 domains, that indicates an exact match. Values less than approximately 3 or 4, depending on the length of your sequence, are very good for per domain hits. So moving on from the best hits section, we then have a section called alternate hits, where we list, instead of just the top hit for each domain, we list the top 10 hits per domain. The information about the query domain appears at the top of each section, and there's a button that allows you to highlight that domain in the structure representation shown above, in the 3D structure shown above. So we list this information for each of the query domains, and we list 10 hits per domain. And the information in these tables mirrors what we saw for the best hits. We have the domain ID from CATH. In this case, we have the superfamily from which that domain is taken, and the PDB entry from which that domain is taken, and the rest of the metrics as described for the best hits. Coming below this section, we have another section called multi-domain matches. So given that we've segmented our query chain into domains, and then we've performed searching on a per domain basis, it's perfectly reasonable to ask, well, are there entire chains in the database that can match all the domains in our query? And this is exactly what the multi-domain chain matches section aims to do. We have a procedure for identifying domain-domain correspondences between the query and database structures, and then we list them in 4 different categories, as I mentioned at the start of this video. The first category is called exact chain matches, where we have the same domains as the query in the same order as the query with no extra domains in the hits relative to the query. And so we can see here that there are a number of such hits. In the table, we list the name of the hit chain. This is a link to the CATH page for that chain. We have a link to the PDB entry from which that chain is taken. We have the total number of domains in the hit chain. And we then have a multi-domain architecture diagram. This is essentially a schematic representation of the domains in the hit. Each of these colored blocks is clickable, and it takes you to a link for the CATH page for the superfamily in question, as we saw previously. So our query structure contains 4 domains, and each of these hits also contains 4 domains in the same order as shown in the query structure. The order of the colors reflects the ordering of the domains in the query. So the first domain is in blue, the second in green, the third in red, and the fourth in magenta. Coming down the list, we see that there are no contiguous or discontiguous chain matches for the query. So that is to say, there are no hits in the database where we have the same domains in the same order, except that there are intervening extra domains in the hits, or domains at either the N or the C terminus in the hit relative to the query. We do, however, have unordered chain matches. These are hits where we have to have the same 4 domains as the query, but they can be present in any order. And as you can see, relative to the hits listed under exact chain matches, the ordering of the magenta and red domains has been reversed. And you can also notice that it's the same chains that are present in both the exact chain matches and the unordered chain matches. And this is a correct result and an expected one. And the reason for this is that the last 2 domains in the query are in fact from the same superfamily. So they are from what's called the immunoglobulin superfamily, or the Ig-like superfamily, if you like. This is a very large, very common superfamily of domains. There are many, many examples of Ig-like domains that perform all sorts of different functions. They have a very conserved structure and so can be aligned with each other at a TM align score of 0.5 or higher very easily. And so if we're thinking about domain-domain correspondences between the query and the hit, and we consider query domains 1, 2, 3, and 4, given that 3 and 4 are essentially copies of the same domain, if I was to map our query chain to one of these hit chains, there are actually multiple ways I could do it. I could do it as 1, 2, 3, 4, but I could also say 1, 2, 4, 3. This would be an equally valid mapping because the terminal 2 domains can be matched either in order or in the opposite order to the last 2 domains of my query. We'll take a look at another example of such a case, and this type of ambiguous matching is a particular problem with repeats of domains. And IG domains frequently occur as repeats in protein structures. And we'll take a look at another example of IG repeats. So here's an example of a query that is composed of two IG-like domains. And as I mentioned, it's very easy to find entries in the PDB that have multiple repeats of IG domains. If you're interested, the PDB code that I've used as the query is one IAR, and we've taken chain B and searched it against CATH. Here we find that the individual domains are matched to the IG superfamily. And looking at the multi-domain chain matches, we find now that we have entries in all four categories. And this is a consequence of the fact that we're dealing with repeats of domains. So our query has two IG domains. We find many examples of hits that have four. And again, you'll find the same hit chain listed in multiple categories. So between the exact chain hits and the unordered chain hits, we have the same chains listed, and that's because mappings 1122 and 1221 are equally valid. So that's the exact chain and the unordered chain matches. Looking at the contiguous chain matches, here we find that we have hit chains that have four domains in them, and the same hit chain is listed more than once in this category of matches, and the same chain is also listed under discontiguous chain matches. This again is a consequence of the fact that the hit chain in this instance is comprised of four IgE-like domains. And so taking the two domains in our query, we can match them to domains 1 and 2, or 2 and 3, or 3 and 4 of the hit chain. And when they're matched in this fashion, they're categorized as contiguous chain matches because domains 1 and 2 of the query have been matched without an intervening domain between them. Looking at the discontiguous chain matches, the same domains can also be matched to the same hit chains in different fashions. So domain 1 of our query matches domain 1 in the hit, domain 2 in our query matches domain 3 in the hit, or domain 4, etc. So there are multiple mappings possible between the query and the hit, and when such ambiguous mapping is present, Merizo-search will show us all such possible matchings. And this again is just something to be careful of when you're dealing with queries that are composed of repeats of domains. The Merizo-search results pages look slightly different if you're searching your query against the TED database as opposed to the CATH database. Here I've shown the results for a chain taken from the PDB, one OPK, chain A, searched against the TED database. This chain has four domains. So similar to what we saw for the previous queries, the domains are listed along with various properties of the top hit. Here, instead of hit CATH, we now have hit TED, and so we are listing the domain from the TED database that matches the query domain in question. The link here will take you to the TED website. So this is a website for the TED resource, and that particular domain matched TED02 in this chain, so we can highlight that. So it shows the AlphaFold database structure with the domains identified by the TED resource colored in. And there's additional information about the domain in question, which I won't go into too much detail about, but you can read more about that in the TED paper and on the TED website. So in addition to listing the hit, for TED, we have the facility to download the model of the TED domain in question. So this would be the hit domain that matches a particular domain in your query. So this again is retrieved from the TED database resource, and it's completely optional whether you want to download this or not. But if you're interested in viewing that domain in isolation, you can download it using these links. Most of the TED domains have been assigned or mapped to CATH H families, and where that has been done, we list that H family along with a link to that superfamily listing on the CATH website. And then we have much the same information as we've seen previously, cosine similarity, etc. And at the end of this table, we also have a link to the species from which that AlphaFold database entry was taken. This will be a link to the NCBI taxonomy browser page for that particular tax ID. And again, I won't go into details about what information's available here. That's information that you can find on the NCBI pages. But it's a nice, convenient way of having information about, well, what species contain these types of domains, what species contain the proteins that I'm interested in. It's important to note here that not all domains in TED will have CATH H families listed up to the fourth number. As we've seen previously, CATH lists the numbers as C.A.T.H. In some cases, we were only able to assign TED domains to the T level, in which case this number will only have three parts rather than four. And in some cases, no assignment could be made at all, in which case there will simply be a dash in this column. So looking down the list at the alternate hits, we see a similar sort of organization where we have the TED hit domain listed, as long as the link to get its all-atom PDB file. And then we have the H family and the other properties of these hit domains. Scrolling down further, we see the multi-domain chain matches section, and analogous to what we saw for the CATH version of this output, we have links to TED entries. So this will be a link to the TED website entry for that chain that we think hits our query. We have a link to the AFDB entry for that chain. So this will just take you to the AlphaFold structured database website for that particular chain. And it also lists the total number of domains in the hit chain according to TED in this case. And we can see that we have hits again in all four categories, and some of these hits have additional domains relative to our query, which is quite interesting. And just for reference, I ran the same query against the CATH database, and you'll see that looking at the multi-domain chain matches, for example, we only have hits in the exact chain matches category, and this suggests that there's quite a lot more information in the AFDB or the TED resource as compared to CATH. And this is something that you should consider when running your results. If you're not getting a lot of interesting results from just the CATH database, then it could be worth also running a search on the AFDB or the TED database. Lastly, let's take a look at the various result files that you can download from a Merizo-search job. They can all be accessed in this panel here. You can download them individually, or, and this is the case for any predictor that you run on the PSIPRED web server, you can download all of them in a single zip file. I've already downloaded this file, and I've got the contents open here in VS Code, but you can use an editor of your choice. I have a little plugin that formats files produced by the method so that the columns line up, but you don't have to have that. This is just for convenience. So let's go through these files one by one. The segment.tsv file and all the files that have the word Merizo in their file name are outputs from the segmentation step using Merizo. So these correspond to just the part of the program that chops up your query structure into domains. So we'll start with the segment.tsv file. The columns are file name, so this is the input file name. You'll find this long string in all of our output files. It's a UUID that's used to uniquely identify your job on the web server. nres stands for the number of residues in your query structure. nres-dom means how many residues out of those were in domains identified by Merizo. nres-ndr stands for the number of residues in non-domain regions, and there are only 11 in this particular job. ndom stands for the number of identified domains. piou means the predicted intersect over union score. This is also sometimes called the Jacquard index, but essentially it's a measure of Merizo's confidence in the segmentation that it's produced. Values above 0.75 are considered good, and so this is a fairly confident prediction. Runtime is listed in seconds for the segmentation step. And then finally, we have the result column, which lists the chopping string that we like to call it, that describes the residue ranges for each of the identified domains. Each domain is separated by a comma, so you can see there are 4 such residue ranges in this particular result, and the individual domain residues are separated by a dash. Where you have missing residues in your input structure or you have discontinuous domains, there will also be an underscore that separates the so-called subranges of the chopping. So if, for example, my first domain was comprised of 2 segments, it might look something like 83 to 90 underscore 100 to 139, say. and that would be a discontinuous range. But in this case, all of the domains are continuous, and so we don't have that. All right, let's look at the next one. So we'll look at search.tsv. This is an output from the single domain search step in Merizo. So after segmentation, we then take each of the identified domains and search them individually against the database. And so this list results for each of the identified domains, and each line in this file corresponds to a single query domain target domain pairing, if you like, or a hit. So if we go down this list, you'll see that there are then results for Merizo_02, which means it's the second domain identified from our structure. So let's go through the columns of this file. The first column is query, which lists the name of the identified domain, the chopping string for that domain. DOMConf stands for domain confidence, and this is Merizo's confidence value for that specific domain. And again, values above 0.5 should be considered good. DOM PLDDT is the average PLDDT of the residues in that domain. And as I mentioned previously, if your query structure is taken from the PDB, this will in fact be a B factor. So you should remember that it's only PLDDT if the query structure is an AlphaFold structure. Then we have mrank, which is embedding rank. So this is the rank of this particular hit based on this column, the embedding score. The highest score will have rank 0, the next highest will have rank 1, etc. Then we have target, which is the name of the domain in the database that was matched. And then we have a number of metrics that are output by TM-align. So we have query length, which is the length of the query domain, the TLen, which is the length of the target domain or the hit domain, alignment length, which is the length of the alignment between these domains, sequence ID, which is the sequence identity reported by TM-align for this alignment. And then we have the two TM scores, query TM and target TM. And this column, max TM, simply reports the larger of these two values. In this particular instance, they're the same, and so you get the same number here. And then we have RMSD. So much of this information we've already seen on the website pages, but here we have information available for up to 100 hits per domain, whereas on the web server we only show 10 on the web server page. The last column is called metadata, and this refers to essentially various annotations that we have for the target domain that was identified. This column is in a format called JSON, and it's a way of essentially encoding structured data in free text. So if you want to look at the format and what this means, you can find that information on the GitHub page for Merizo-search. I won't go into detail here. The other reason we have this in JSON format is that it allows for flexibility in the specification of exactly what information is in this column. And this information will differ between CATH and TED, whether you're using the CATH or the TED database. And again, that information about the format can be found on our GitHub site. We'll then look at search multi-DOM. So this will contain the results of the multi-domain searching step. As you recall, this particular query structure only generated hits that were classified as exact chain matches. So we only have a few results here. If we had hits in other categories, they would be listed underneath these. So again, we have query chain. This time it's referring to the entire chain that you have provided. So it's important to remember that multi-dom.tsv refers to complete chain results, whereas search.tsv refers to per domain results. So here we have the query chain listed, NQD, which stands for the number of query domains, so the number of domains in the query structure. We have hit chain, which is the name of the chain from the database that we were able to match. NHD, which stands for number of hit domains, so the number of domains in the hit chain. Match category, which is a number from 0 to 3. It's an integer, 0, 1, 2, or 3, which describes whether this is an exact MDA match, an exact multi-domain architecture match, or one of the other three categories. So match category 3 corresponds to an exact MDA match, and match category 0 corresponds to an unordered domain match. The next column is called match info, and this gives us the information about the pairings between the query domains and the hit domains. So what we have here are, first of all, commas separating the individual domains. And then within each of these units, we have three values separated by colons. So the first value will be the name of the query domain. The next value will be the name of the hit domain. So here you can see that query domain 1 has been matched to hit domain 1. And then the last entry within that unit will be the maximum TM score of the maximum of the query TM score and the target TM score for this particular domain pair. So then we have a comma, and then it lists the pairing for the second domain. And you can see that that's matched against the second domain of the hit with this TM score, and so on. Finally, we have hit metadata, which is again a JSON string, but this time it is a JSON array. And this has the metadata taken from the database for the hit domains, so these ones, in the order that they appear on this in the match info field. So that is again quite useful when we're trying to automate certain annotations. We use this information when we're making the multi-domain architecture diagrams on the web server. All right, so let's look at search insignificant.tsv. This once again refers to the single domain matching step. And in this file, we would list any hit domains that don't pass the 0.5 TM score threshold that I mentioned earlier. In our case, we say that the method is allowed to match up to 100 domains per query domain. And in this particular instance, all 100 of them have a TM score greater than 0.5. And so this file is empty. And this is actually quite a good result. Let's take a look then at Merizo.pdb2. This is a PDB formatted file. It has only atom records in it. And essentially what it is your query structure, but the occupancy column here has been modified to include essentially identify the domains. And this is really useful if you're using a structure viewer because it lets you color the structure according to the domains. So let's take a look at how that might be done. I've got PyMol open here on the left. So we'll just drag this file into PyMol. And to color the structure by the occupancy field, we would type spectrum Q. And so now it has colored the structure according to the domains identified by Merizo. And the dark blue section here corresponds to the non-domain residues of this particular structure. This shade of dark blue is used by default for the non-domain regions, but you can easily change that by saying color gray, let's say, and then just say Q less than 1. So all the NDR residues are assigned a value of an occupancy value of 0. Okay, so let's go back and look at the next file. Merizo.idx is simply a mapping from the PDB residue numbers to the domain ID identified by Merizo. So for each residue, you have a colon separation between the residue number and the domain number. And as you move along this file, you can see you've got the start of domain 2 and so on. And finally, non-domain residues, as I mentioned, are indicated with a 0. Then we have 4 DOM PDB files. These again are PDB formatted files, but they are essentially just chopped out of this one, one for each of the identified domains. And again, you can import these into a structure viewer such as PyMol. It may not recognize that extension, so you might have to manually type something like load this dom.pdb and say format equals PDB. And then you can see the structure of the individual domain. And you can repeat that for each of the DOM PDB files. And there will be a variable number of these according to how many domains were identified. And then with all of our jobs on the server, you have this job configuration.csv file, and that simply tells you the versions and the names of the software packages that were used for your job, as well as the parameters that were passed to it. So if you were to run Merizo-search on your own computer or on a local cluster, for example, these would be the parameters that you would use to replicate what you see on the server. Okay, I think that about covers all of the outputs from Merizo-search. And as always, if you have any questions, please don't hesitate to get in touch with us. Thank you for watching.